Today's lesson is on the endomembrane system. This includes the endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, vacuoles, and vesicles. All of these together play a key role in the synthesis and hydrolysis of macromolecules inside the cell. First up is the endoplasmic reticulum. Its main function is to manufacture membranes and perform many biosynthesis functions. Remember, synthesis is to build, so we're building molecules. The structure is a gigantic membrane that is connected to the outside of the nucleus, the nuclear envelope, and it extends throughout the cell. It accounts for about 50% of the membranes in all eukaryotic cells. And you can see here in this diagram, it is this blue kind of zigzaggy membrane filled with different pockets. Some of the endoplasmic reticulum is labeled as rough endoplasmic reticulum because it contains bound ribosomes on it, and you can see the little speckly dots there. And then there are other sections of the endoplasmic reticulum that are considered smooth because they have no ribosomes attached to them. And you can see that in this diagram here. You have the rough endoplasmic reticulum and then the smooth endoplasmic reticulum. So structurally, they're a little different. You can see how the rough ER is more kind of these long chambers here, right? These long chambers. And then the smooth endoplasmic reticulum is more like little bubbles almost. The function of the smooth endoplasmic reticulum is to synthesize lipids, oils, phospholipids, steroids, and hormones. Remember, those are all in the lipid family. It is also responsible for the breakdown of glycogen into glucose in the liver, and it also has detoxifying responsibilities for drugs and poisons in the liver. So there's a lot of smooth endoplasmic reticulum in the liver cells and anywhere that needs to synthesize a lot of lipids. The rough endoplasmic reticulum is primarily for the production of proteins that are going to be exported out of the cell. Because of the ribosomes that are located on the rough ER and the endoplasmic reticulum makes all those membranes, those proteins that are made get packaged into that membrane right there, and then they get transported to the next step of the process. So the endoplasmic reticulum, part of it is responsible for making membranes, right? Making those phospholipids. Again, lipids. Endoplasmic reticulum builds new membranes. As the endoplasmic reticulum membrane expands, it buds off, and then they transfer to other parts of the cell that need those particular membranes. And then the same thing happens when they synthesize membrane proteins. The proteins are embedded in that membrane, and then eventually they will become part of the membrane. So it's pretty convenient if you want to make a protein that's going to end up in the cell membrane to package it in something that is also made of membranes. And you can see the process here in the diagram. Make the membranes, make the little proteins, package them, and then they get processed for export out of the cell. So the next step in our process here is for these little pieces of membrane to go to the Golgi apparatus. The function of the Golgi is to finish whatever molecules the endoplasmic reticulum has produced, to sort them, and to kind of determine where they're going to be shipped out to. So it's kind of like a shipping and receiving department in a factory. They are the center of the manufacturing, warehousing, sorting, and shipping, and they are very extensive in cells that are specialized for secretion of molecules outside of the cell. The structure of the Golgi, they are flattened membranous sacs. Remember, this is the endomembrane system, so all of these things are made of membrane. One side of it is the cis side, that's the entrance, and then the trans side is the exit. This is where the little vesicles are kind of bud off for transport. Those flattened sacs are called cisternae, and they kind of do look like flattened little pockets of pita bread, if you know what pita bread is. Here's another diagram of the Golgi and what it actually looks like inside of a cell. So products from the endoplasmic reticulum enter on the cis side of the Golgi. They are modified into their final form. They are tagged for distribution wherever they're headed, and they are packaged into little transport vesicles, which come out on the trans side of the Golgi. So when we put these pieces together, we have information that comes from the nucleus to go here, the instructions to make those proteins, and then the ribosome makes those proteins. The endoplasmic reticular makes the membrane, puts them in this little package here, brings it over to the Golgi where it is processed and tagged for transport, and then they get transported in these little vesicles. Another component of the endomembrane system are lysosomes. Lysosomes are membrane-bound sacs filled with hydrolytic enzymes that digest macromolecules. So these are very important in the process of hydrolysis, right? How we break our macromolecules into its individual monomers. So they're like a little stomach for the cell. Lysosome means to break things apart. And they're also considered the cleanup crew of the cell. So when other organelles or things are broken, they actually get consumed by a lysosome, and the lysosome will break them down. Okay, so they're like a little garbage can as well. And they are only found in animal cells. So the lysosomes fuse with a food vacuole. So you have some food that's going into the cell, and then it fuses with the lysosome here, and then the lysosome's enzymes will break it down. Another function is for a broken piece of a molecule to fuse with a lysosome, and then the lysosome will also break that down so that all of these things can be recycled. So here's another look at that. Same process, food vacuoles fusing with a lysosome for digestion, or an old molecule. In this case, it's the mitochondria that is damaged, and it's going to be broken down for digestion. So the lysosomes are very interesting because they digest things inside the cell. So this organelle has evolved to have a custom pH. Again, think about what you know at enzymes. Enzymes work best at a particular pH. And in this case, it's a pH of 5. Why do you think it's a pH of 5? Because the rest of the cell has a pH around 7. So if those enzymes happen to break out of a lysosome, what will happen to them? If they work best at pH of 5 and they escape out into the rest of the cell, will they work anymore? No, they've been denatured, right? The change in pH will affect the shape of those lysosomal enzymes and will cause them to denature. That way the cell's entire contents are not digested if a lysosome happens to break and those enzymes escape into the rest of the cell. There are some things that can go wrong. When a lysosome's digestive enzyme doesn't function properly, then you can't digest biomolecules. So instead, those macromolecules collect and they get filled up with undigested material. So an example of this is a disease called Tay-Sachs disease, where lipids build up in brain cells because the lysosomes don't function to break them down, and it usually results in a pretty early death for the child, unfortunately. Sometimes it's supposed to work that way. Sometimes cells are actually supposed to die. It's called apoptosis, and it's called programmed cell death or programmed destruction of cells. There are some important things that why this has to occur. One of those is actually when you are developing. So when you are a couple of months old, you actually have like a little mitt. Your fingers are all fused together. You can see that here in that picture. And then programmed cell death actually kills the cells that are in between your fingers, causing that to go away. There are times where it doesn't work properly, right? And we get fingers or toes that are fused together in an individual. Another type of digestive enzyme little container is called a peroxisome. These are the organelles that contain the enzyme that breaks down peroxide, right? We know that peroxide is deadly to cells, H2O2, as we talked about in our lab. And those digestive enzymes will break down the fatty acids and sugars, but they produce peroxide as a byproduct. So your cells must have these little peroxisomes, this particular digestive enzyme sac that contains the right enzyme, that's catalase, to break down that hydrogen peroxide and turn it into water so that it does not kill other things inside your cells. Vacuoles and vesicles are almost like little storage containers. Some food vacuoles will fuse with lysosomes and will have those enzymes in them to be able to digest like a lysosome. Some of them contain water. There are vacuoles in plants that contain water because plants need water to keep their cells large and for all of their general functions. Vacuoles are generally just found in plants. They control different things within the cell, but most of them are for storage of water. So vacuoles are just in plants. You can see a large central vacuole right there, filled with water. Vesicles are smaller, and they are mostly found in animal cells. And when we put all that together, again, you can see the process here. And here are some final thoughts.